Hello everybody, welcome to my channel Omni Bio Entrance, and today we will be discussing IIT JAM question paper for the subject Biotechnology conducted in the year 2016. I will be discussing MCQ, MSQ and numerical aptitude test from biotechnology in this question paper. If you are new to my channel, please do like, share and subscribe. If you want previous year question papers of IIT JAM, you can go to my channel Omni Bio Entrance. In the playlist, search for IIT JAM, you will get it there. Coming to the first question. Pure IgG antibody was run on an SDS page under reducing condition. How many bands would you see after staining with Kumasi blue? So IgG is having two heavy chains and two light chains which are joined by disulfide bonds. Here they have specifically mentioned that it is under reducing conditions that you run the SDS page. So under reducing condition means you use beta mercaptoethanol or dithiotritol to break the disulfide bonds present between the light and the heavy chains of the IgG molecule. So you will get two bands after breaking the disulfide bonds, that is heavy chain at 50 kDa and light chain around 25 kDa. So the answer here is option B, 2. Then next one. During mitosis, disappearance of the nucleolus is a hallmark of. So it is a hallmark of the prophase stage. In the prophase stage, chromatin condenses, nucleolus disappears, nuclear envelopes break down and centrioles separate. Next one. Eukaryotic cell containing flagella is cell lining the fallopian tube. It is of cilia, not flagella. Paramecium is also having cilia. Cells lining respiratory tract also has cilia. Sperm is having flagella. Sperm is having flagella so that it can move through the fluid and reach the egg. The circulatory levels of estrogen is derived mainly from. So circulatory level of estrogen, it refers to the concentration of estrogen present in the bloodstream. That is called as circulatory level of estrogen. So you use two cells for the derival of circulatory level of estrogen, that is theca cells in the ovary and granulosa cells in the ovary. So answer here is option A. Then next one. Radial symmetry is the characteristic feature of which of the following phyla? Arthropoda is having bilateral symmetry. Mollusca is also having bilateral symmetry. Cnidaria is having radial symmetry. Chordata also bilateral symmetry. So your answer is Cnidaria. In ecotone, some species become abundant and they are called. So what is an ecotone? It is a transitional zone between two or more distinct ecological communities. That is, a transitional zone will be present between a terrestrial and aquatic ecosystem. That is called as ecotone. Okay. Now option A, they have mentioned as sibling species. They are, what are sibling species? They may be two or more species which are closely related to each other. They are derived from the same common ancestor. They have minimal morphological differences. They occupy similar ecological niches and they are reproductively isolated. They are called sibling species. So it has nothing to do with ecotone. Then endemic species means they are plants or animals that are found in a specific geographic region. They are found nowhere else other than in this specific geographic region. That is pandas in China, Galapagos giant tortoises, all these are endemic species. Then rare species they are smaller in population size and are found in limited geographic areas. And they are vulnerable to extinction also. These are called as rare species. So what remains? Edge species. Some species become abundant and they are called edge species. Answer is option D. Then next one, match the entries in group 1 with entries in group 2. Epilepsy. Epilepsy is caused due to defect in the electrical discharge in the neurons. So P4. Alzheimer's disease is due to decreased production of acetylcholine. Q3. Parkinson's disease is due to degeneration of dopamine releasing neurons. And Huntington's disease is due to degeneration of neurons in the cerebral cortex. So P4, Q3, R2 and S1 gives the answer as option B. Then determine the correctness or otherwise of the following assertion and reason. Assertion. In the process of ATP synthesis in oxidative phosphorylation, okay, ATP synthase is not part of electron transport chain. Yes, it is not a part of the electron transport chain of inner mitochondrial membrane. Electron transport chain, it consists of series of protein complexes, that is complex 1, 2, 3, 4 and 5 in the inner mitochondrial membrane, and they transfer electrons from high energy molecules to oxygen, and thereby they will generate a proton gradient across the membrane. That is what is happening in electron transport chain. But ATP synthase is coupled to electron transport chain through proton motive force. So ATP synthase, it is embedded in the inner mitochondrial membrane. That's true. But it is not a direct part of electron transport chain. It uses the proton gradient generated by the electron transport chain, and it will drive ATP synthesis from ADP and Pi. It is coupled to electron transport chain through proton motive force. So here both A and R, that is assertion and reason, are true, and R is the correct explanation of A. So the answer here is option A. Then next one, higher levels of glycosylated hemoglobin HbA1c indicate. So HbA1c, it refers to glucose attached to hemoglobin in the red blood cells, and it is a blood test measuring average blood glucose over 2 to 3 months. Okay, so higher HbA1c, it will indicate that the person is having diabetes. Answer is option C. Match group 1 with entries in group 2. DNA replication. DNA replication experiments were conducted by Michelson and Stahl. P3. Genetic code was discovered by Nirenberg and Corona. Life on Earth was by Miller and Urey experiment. DNA as genetic material was proved by Hershey and Chase. So P3, Q4, R2 and S1. Answer is option C. Then next one, again match the following. Bacteria. Bacterial disease out of the given in the group 2 is anthrax. So P2. Then virus disease is rubella. Q3. Fungal disease is athlete's foot, and protozoan disease is leishmania. S1. So P2, Q3, R4, S1 is option A. Then next one. In an experiment conducted in the dark, isolated chloroplasts are kept in buffer pH 4 at 4 degrees Celsius until their internal pH is equal to 4. Okay, so they are isolated the chloroplast, kept in the dark, kept in a buffer of pH 4 at 4 degrees Celsius. So that they are kept in that buffer solution until the internal pH of the chloroplast is also equal to 4. Then they are transferred to a buffer of pH 8. So these chloroplasts which have the internal pH of 4 are transferred to a buffer of pH 8, and you add and give ADP and Pi also. What will happen? So first, At pH 4 degrees Celsius, chloroplasts are in an inactive state. At pH 4 degrees Celsius, chloroplasts are in an inactive state. Now they are transferred to pH 8 buffer, and this creates a proton gradient across the thylakoid membrane. And there they have added ADP and PI also. So what will result because of this proton gradient? ATP synthesis will be the outcome. Okay. So chloroplast will be destroyed. Option A. They will not be destroyed because the conditions are not very harsh. So they will not be destroyed. Chlorophyll in the chloroplast will release bound magnesium. That is also not possible. Chloroplast will be intact but no ATP. ATP will be produced. Chloroplast will be intact and ATP will be produced. Answer is option D. Then next one. Two mammalian cell lines with doubling times of 12 hours and 36 hours were cultured with radioactive thymidine for 8 hours. The cells were further cultured without radioactive thymidine for 72 hours. Incorporated radioactivity was measured in equal number of cells in each culture, which revealed that both cell lines had the same amount of radioactivity. No, one is fast growing, one is slow growing. The slow growing is the one at 36 hours and fast growing is the one at 12 hours. Fast growing cells have more radioactivity. Fast growing cells have shorter rest phases, so they will not have more radioactivity. Slow growing cells have more radioactivity because they have longer rest phase. Slow growing cells spend more time in the G1 phase, and once they reach the S phase, they incorporate more radioactive thymidine in them due to longer rest phase in slow growing cells. So the answer here is option C. Then match the following. Yeast hybrid system is used for. They have given some techniques in group 1 and you have in group 2 what they are used for. So yeast 2 hybrid system is for protein-protein interaction studies. P4. Electrophoretic mobility shift assay is for in vitro protein DNA interaction studies. Chromatin immunoprecipitation is for in vivo protein DNA interaction studies. Nuclear magnetic resonance or NMR is for protein structure determination. So P4, Q3, R1 and S2 gives the answer as option B. Then comes to multiple select questions. Identify the autoimmune disease among the following. So there two options in this question are correct. So type 1 diabetes mellitus and pernicious anemia. They both are autoimmune diseases. Type 1 diabetes mellitus, that is the autoimmune destruction of the beta cells of the pancreas. That is type 1 diabetic mellitus. Pernicious anemia is destruction of gastric parietal cells. And these gastric parietal cells, they produce intrinsic factor. Now upon the destruction of these gastric parietal cells, intrinsic factor production is reduced. Reduction in intrinsic factor production means there is less absorption of vitamin B12. Vitamin B12 deficiency can lead to pernicious anemia. So answer here is B and D. Then next one. Which of the following statements are true for hydrogen bonds? Strength of hydrogen bond is low in a solvent of high dielectric constant. Yes. Low in a solvent of low dielectric constant. No. Lower in water as compared to organic solvents. That is also correct. So A and C are correct. Then which of the following statements are true for cellulose? Cellulose serve as structural role. That's correct. Cellulose is a branched polysaccharide. No, cellulose is an unbranched polysaccharide. So B is not correct. Cellulose is a homopolysaccharide composed of alpha-1,4 linked D-glucose. No, it is beta-1,4 linked D-glucose units. So cellulose is a homopolysaccharide composed of beta-1,4 linked D-glucose units. D is the correct answer. Now next one. Which of the following are not true for photosynthesis? They have asked not true. Reduction of carbon dioxide and water. No, that is not correct. Oxidation of carbon dioxide and water. Again no. Reduction of carbon dioxide and oxidation of water. That is what is happening in photosynthesis. Oxidation of carbon dioxide and reduction of water. No. So A, B, D are the wrong answers. Then next one. Apoptosis is controlled process of cell death. Yes, it is programmed cell death. The process involves exposure to phosphatidyl serine on the outer surface of the cell membrane. Yes, that happens during apoptosis. Decreased permeability of the outer mitochondrial membrane. No. It involves increased permeability of the outer mitochondrial membrane, and increased permeability allows cytochrome C and other pro-apoptotic factors to escape from mitochondria. Increased lysosomal activity. No. Internucleosomal cleavage of genomic DNA. That is also correct. So A and D are correct. Now coming to NAT, that is numerical answer type questions. The first one, the net charge on the following peptide at pH 7 is. So they have given the peptide valine, aspartic acid, asparagine, lysine, serine, isoleucine. So this is the peptide. I have drawn it here. This end will be the N terminal end and it will have N3. And this will be the C terminal and it will have COH. N3 will have a +1 charge and COH at -1 charge. Aspartic acid and lysine, they will have side groups. So here aspartic acid will be having COH side group, lysine will be having an NH3 side group. At pH 7, it will be -1 for aspartic acid and +1 for lysine. Therefore, the net charge is -1 plus +1, that is equal to 0. Then next one. A 152 nanometer long Watson-Crick double helical DNA, bDNA will contain how many turns? So, number of turns is equal to length of DNA by pitch of the DNA. So, length of the DNA they have given 152 nanometers. So, substitute that. Pitch of the DNA, it is bDNA. Pitch of bDNA is 3.4 nanometers. That will give you 44.7. But number of turns cannot be a fraction. So, it will be round off and you can write it as 41 and 44. Then next one. A population is in Hardy-Weinberg equilibrium for a gene with only two alleles, A and small a. If the gene frequency of allele capital A is 0.7, the genotype frequency of heterozygous capital A small a is. So P value they have given, that is the gene frequency of allele capital A, it is 0.7. From there you can find gene frequency of allele small a. That is Q. That is because of the relation P plus Q is equal to 1. So Q is equal to 1 minus 0.7, that is 0.3. Now genotype frequency, there is a formula P square plus Q square plus 2PQ is equal to 1. What is P square? The genotypic frequency of capital A. That is 0.7 square is equal to 0.49. For Q square, it will be small a, it will be 0.3 square, that is 0.09. But in the question, they have asked only the genotype frequency of heterozygous condition, capital A, small a. So, capital A, small a will be 2PQ. That will be the heterozygous condition. So, 2 into 0.7 into 0.3, that is 0.42. So, genotypic frequency of heterozygous capital A, small a is 0.42 or 42%. Then next one. A receptor binds to its ligand with the dissociation constant of 10 raised to minus 8. The concentration of the ligand required to occupy 10% of the receptors would be 10 raised to minus x molar. The value of x is. So there is a formula: bound receptor by total receptor is equal to ligand by concentration of ligand by concentration of ligand plus Kd, where Kd is the dissociation constant. Now for 10% occupancy, bound receptors will be 10, total receptor concentration will be 100, that is 10%, is equal to concentration of ligand by concentration of ligand plus 10 raised to minus 8. So 0.1 will be concentration of ligand by concentration of ligand plus 10 raised to minus 8. Solve for concentration of ligand. These are the steps that are involved in solving. So you will get concentration of ligand is equal to 0.1 into 10 raised to minus 8 by 0.9, or that is equal to 1.11 into 10 raised to minus 9, or approximately 10 raised to minus 9. They have given the value of concentration of ligand is 10 raised to minus x molar. So 10 raised to minus x is equal to 10 raised to minus 9, or x is equal to 9. Then next one. An enzyme catalyzes the conversion of 4 into 10 raised to minus 4 molar substrate into product at a rate of 20 micromolar per minute. If the Km value of the enzyme is 2 into 10 raised to minus 4 molar, the value of Vmax is in micromoles per minute, molar per minute. So we know the Michaelis-Menten law equation. V is equal to Vmax concentration of S by Km plus concentration of X. So V they have given 20 micromole per minute. Concentration of S, substrate concentration is 4 into 10 raised to minus 4 molar or 400 micromolar. Km value is 2 into 10 raised to minus 4 molar or 200 micromolar. Answer should be in micromolar, that is why I am converting it then and there. So now we have to find out the Vmax value. So this is the formula, I have split it. That's all. And rewritten the formula. Vmax is equal to V into Km plus concentration of S by concentration of S. Substitute the values, you will get it as 30 micromolar per minute. Then next one. The following polypeptide chain was sequentially treated with dithiotritol and cyanogen bromide and trypsin. So this is the polypeptide chain. The total number of fragments expected at the end of complete digestion of the polypeptide. Consider that none of the reagents interfere with each other's activities. Okay. Now cyanogen bromide, it cleaves the carboxyl side of methionine. So here you have got a methionine, a cut over there. Here is another methionine, a cut over there. Trypsin will cleave carboxyl side of lysine and arginine. So lysine cut here, then arginine cut here. So how many fragments you have? 1, 2, 3, 4 and 5. So 5 fragments will be obtained. Then next one. In maize, the genes for colored seed and round seed are dominant over the genes for colorless seed and shrunken seed. Pure breeding strains of the double dominant variety were crossed with the double recessive variety, and a test cross of the F1 generation produced the following. Colored round seeds 380, colorless shrunken seed 396, colored shrunken seed 14 and colorless round seeds 10. From the above, distance between the genes for seed color and seed shape on the chromosome would be dash centimorgan units. So these values, that is 380, 396, they are more than these values. They are the topmost in all the values given here. So 380, 396 will be your parentals. 14 and 10 will be your recombinants. Now recombinant frequency you have to calculate. That will give the distance between the genes for seed color and seed shape. So recombinant frequency is equal to number of recombinants by total into 100. So 14 plus 10 will be number of recombinants, that is 24. Total will be total progeny, that is 380 plus 396 plus 14 plus 10 will give you 800 into 100 will give you 3 centimorgan. Then next one. A culture of 10 raised to 6 bacteria with a doubling time of 60 minutes is grown in a nutrient medium at 37 degrees Celsius. Considering that the nutrients are unlimited, the number of bacteria at the end of 10 hours would be dash into 10 raised to 6. So 1 hour, 1 generation of bacteria are produced. So 10 hours you will get 10 generations. Okay. Now there is a formula Nt is equal to N0 into 2 raised to N, where Nt is the population after time t and N0 is the initial population and N is the number of generation. Number of generation we already calculated, it is after 10 hours it will be 10 generations. So substitute. Now Nt is population after 10 hours. That you have to calculate. So 10 raised to 6 into 10 into 2 raised to 10. Sorry, it is 2 raised to 10. Okay, the writing is not correct. That's why. 10 raised to 6 into 2 raised to 10. So 10 raised to 6 into 1024. 2 raised to 10 is 1024. So 1024 into 10 raised to 6 will be the answer. Then next one. A 50 amino acid residue stretch of a globular protein adopts an extended structure containing a true alpha helix of 24 residues and beta strand of 26 residues. Okay, so 50 amino acid residue stretch of globular proteins, it is having alpha helix and beta strands of 24 and 26 residues. The total length of the stretch will be. So for alpha helix, we will see what is the total length occupied by the alpha helix. Given 24 residues have given. So length per residue for alpha helix is 1.5 Armstrong. So total length will be 24 into 1.5 Armstrong or 3.6 nanometers. Answer is in nanometers. So convert it then and there. 3.6 nanometers. Now beta strand. Total length we have to see for beta strand. It is given, it is 26 residues are in beta strand. So length for this extended beta strand will be 3.5 Armstrong per residue. So total length will be 26 into 3.5 Armstrong, that is 91 Armstrong or 9.1 nanometers. So total length of the whole stretch plus alpha helix plus beta strand. So 3.6 nanometer plus 9.1 nanometer, will give you 12.7 nanometers. So that's it. Thank you for watching my channel. Please do like, share and subscribe. If you want still previous year question papers of IIT JAM, you can go to my channel Omni Bio Entrance. See the playlist. In the playlist C for IIT JAM, you will get it there. I have solved and explained everything. So thank you for watching.